Look at these spherical virus particles. They are highly organized, repetitive, and completely rigid, but they aren't alive. Viruses are acellular. They lack the internal biological machinery required to live or reproduce on their own. They have to hijack another organism's equipment to survive. To find a true living cell, we have to look at prokaryotes, like these bacteria. A bacterium is fully capable of independent life, but functionally it operates as one single open room. Inside that room, everything shares the exact same fluid space. The bacterium's genetic instructions, its circular DNA, floats freely alongside small 70S ribosomes, which are the tiny structures responsible for building proteins. This single-room design works perfectly for microscopic organisms, but it hits a strict biological limit. If that open cell grows too large, the hundreds of different chemical reactions taking place inside it begin to mix and interfere with one another. The resulting chemical chaos slows down efficiency, making it impossible to support complex life. The evolutionary solution to that size limit is the eukaryotic cell. As you can see here, eukaryotic cells solve the space problem by building internal walls. A eukaryotic cell is significantly larger than a bacterium. To manage that extra volume, it relies on membrane-bound compartments to isolate specific chemical reactions, turning a chaotic soup of cytoplasm into a highly efficient factory. The command center of this factory is the nucleus. Think of it as a secure central vault, surrounded by a double-membrane nuclear envelope. This thick boundary isolates and protects the cell's master genetic code from the busy environment outside. That genetic code consists of roughly 2 meters of linear DNA. To fit all that data inside a microscopic vault, the DNA is tightly wrapped around specific proteins called histones. Together, the DNA and histones form a compact coiled material called chromatin. Deep inside the nucleus is a dark, dense core known as the nucleolus. This is a specialized manufacturing zone where the cell builds its own production machines, the ribosomes. Because eukaryotic cells are larger and more complex, they produce larger ADS ribosomes. Once built, these ribosomes exit the vault through small gaps called nuclear pores and anchor themselves directly onto a folded maze of membranes known as the rough endoplasmic reticulum, or rough ER. The rough ER acts as the primary assembly line. Here, the ribosome translates the genetic instructions into physical proteins. As the protein is built, it drops down into the isolated interior space, the lumen of the ER, where it can safely fold into its final three-dimensional shape. Once folded, the protein is transferred to another membrane compartment called the Golgi apparatus. The Golgi chemically modifies the protein, packages it into a protective bubble called a vesicle, and ships it off to the cell surface. This entire pipeline, from reading the DNA to secreting the finished protein, relies entirely on compartmentalization. By isolating these delicate manufacturing steps, the cell ensures they aren't disrupted by the unpredictable environment of the wider cytoplasm. That wider environment requires constant maintenance. Enter the lysosome, the cellular waste disposal unit. Lysosomes are packed with highly acidic, powerful digestive enzymes capable of breaking down large molecules and damaged cell parts. The membrane surrounding the lysosome is a matter of life or death. If that compartment ruptured, those acidic enzymes would spill out and immediately digest the cell from the inside out. Running this massive walled-off city requires a robust power grid. That role falls to the mitochondria. These compartments perform aerobic respiration, breaking down nutrients to generate ATP, the universal energy currency of the cell. Plant cells have an additional exclusive power plant, the chloroplast. Chloroplasts capture energy from sunlight and use it to manufacture glucose, providing the plant with a storable food source. But there is a biological paradox here. If you look closely at their interiors, you'll see that both mitochondria and chloroplasts possess their own circular DNA and smaller 70S ribosomes. These structures are identical to the ones found in single-room bacteria. This points to the endosymbiotic theory. Millions of years ago, these energy-generating organelles were actually free-living prokaryotes. Early eukaryotic cells swallowed them, but instead of digesting them, they put them to work. Complex life assimilated ancient bacteria, turning former prey into the permanent integrated powerhouses that make advanced cellular architecture possible. Because cells are often drawn as flat circles, it's easy to imagine them as simple bags of fluid. The reality is supported by the cytoskeleton. This is a dynamic web of microtubules and microfilaments that continuously assemble and shift, acting as both transport highways and structural scaffolding throughout the cytoplasm. To organize this shifting web, cells use anchor points called centrioles. These small structures orchestrate the protein filaments, guiding the separation of chromosomes during cell division, or forming exterior structures like swimmy flagella and hair-like cilia. If we zoom all the way out to the absolute border of the cell, we find another rigid structure. Look at these thick, purple-stained hexagonal borders. While animal cells only have a flexible membrane, plant and fungal cells are encased in a solid cell wall made of a tough carbohydrate called cellulose. To maintain their shape, plant cells use a massive compartment called the central vacuole. They fill this vacuole with water and dissolve nutrients, which pushes outward against the rigid cell wall. This creates a strong internal hydrostatic pressure, known as turgor pressure. The combination of outward pressure from the vacuole and the unyielding boundary of the cell wall is exactly how towering organisms, like giant redwood trees, manage to stand completely upright without a single bone in their bodies. Pulling back, we can see the entire eukaryotic cell functioning like a massive living city, with all of these compartments operating simultaneously. The interdependencies are constant. The Golgi apparatus cannot ship its packages without the rough ER. The ER cannot build proteins without the genetic code from the nucleus. And every single step of this process collapses without a steady supply of ATP from the mitochondria. Static, flat diagrams in textbooks make it easy to view organelles as isolated objects floating in empty space. But inside a real cell, it is a highly dynamic, interconnected ecosystem. The leap from simple bacteria to complex plant and animal life required the mastery of specialized space. Understanding biology at this scale requires looking past individual microscopic parts. It means seeing that life thrives on the synchronized architecture of the entire machine. Pulling back, we can see the entire eukaryotic cell functioning like a massive living city, with all of these compartments operating simultaneously. The interdependencies are constant. The Golgi apparatus cannot ship its packages without the rough ER. The ER cannot build proteins without the genetic code from the nucleus. And every single step of this process collapses without a steady supply of ATP from the mitochondria. Static, flat textbook diagrams make it easy to view organelles as isolated objects in empty space. But a real cell is a highly dynamic, interconnected ecosystem. The leap from simple bacteria to complex life required the mastery of specialized space. Understanding biology at this scale requires looking past the individual microscopic parts. It means seeing that life thrives on the flawless, synchronized architecture of the entire machine.